Well, hello everybody, and today we're going to be talking about synapses. So, neurons communicate by transmitting chemicals at junctions called synapses. And we talked about neurons being important for transmitting messages throughout the body, and that there are individual neurons, and they are separated by a small space called a synapse. And we already talked about the idea of an action potential or a nerve impulse that sends a message from one end of the neuron to the other end, right? And that was called the action potential, and that was an electrical signal that was sent down the axon. And now we're going to talk about how neurons communicate with each other, so from one neuron to the next. And initial research was done on reflexes. Reflexes are automatic responses to certain stimuli. And so this early research was started on a reflex arc, and that's a circuit from a sensory neuron to a motor neuron. So here's a sensory neuron in the skin, right? Maybe you touch something that's very hot. A message is sent to the sensory neuron into the spinal cord, and then out to the muscle. And in fact, the first research on reflexes was done using the reflex from pinching a dog's foot, and the dog raises its leg, okay, or flexes its leg. So again, there's the skin or the sensory neuron that's in the skin. The skin gets pinched. There's an action potential that's sent down this sensory neuron, and then to an interneuron, and then out of the spinal cord to a motor neuron. And it was found that reflexes are slower to conduct a message than conduction simply along a single axon. Okay, so we know how long it takes to send a message down an axon, to send an action potential from one end of the axon to the other. And this early research showed that it takes longer to send a message through this reflex arc, okay, which includes a sensory neuron, an intrinsic or an interneuron, and a motor neuron. The speed of conduction along an axon is about 40 meters a second. So remember, we said you send an action potential down a neuron, the sodium channels open, sodium rushes in, potassium channels open, potassium leaves the cell, then the sodium channels close, potassium continues to leave the cell, that's bringing the cell back down to its resting potential of -70, and then that happens again at this part of the axon, and the next part, and the next part. But the speed of conduction through a reflex arc, and now we have a neuron that must send action potentials, and then this neuron C has to communicate with neuron D, which then sends an action potential down its axon. And then that neuron has to communicate with neuron E. And so the speed of conduction through this reflex arc is slower, sometimes 15 meters a second or less. And so the thought is that the delay somehow occurs in these synapses, in this communication area of communication between one neuron and the next. So synaptic transmission is the way neurons communicate with each other, and messages are carried by chemicals called neurotransmitters. We're going to abbreviate that NT. So these neurotransmitters basically diffuse across the fluid-filled synaptic cleft. So that space between the neurons, or the synapse, it's also called a synaptic cleft, is filled with fluid, and these neurotransmitters or chemicals float or diffuse across them from the presynaptic terminal, or the end of one cell, to receptors on the postsynaptic membrane. And we're going to go over a picture that I think will make this all more clear. And these neurotransmitters fit into receptors like a key in a lock, and then turn on the receptor, producing what we call postsynaptic potentials. So that's the idea of the synapse in a nutshell. But let's take a look. Here's a very simplistic sketch of a synapse. So we have a message being sent this way from left to right on your screen, and we have action potentials that would be sent down the axon, okay? And this is the end of the axon, which we call it a presynaptic terminal, meaning the end of the axon. Well, remember, here's the synapse, or the space between the neurons, and so this is now called the presynaptic, right, before the synapse terminal. Now that kind of makes sense as a name for the end of the axon, the presynaptic terminal. Here's the synapse that's filled with fluid, and then this is the dendrite of another cell, and we would call this the postsynaptic cell. So again, here's the synapse, and this is postsynaptic, or after the synapse. So here in the presynaptic terminal, so remember this is the end of the neuron, there are little circles, little balls that contain neurotransmitters, and neurotransmitters are chemicals. And these little balls containing the neurotransmitters are called vesicles. So we have vesicles of neurotransmitters, and they're all kind of sitting here at the end of the neuron at the presynaptic terminal. So the presynaptic terminal is the end of the presynaptic neuron. The postsynaptic neuron, that's the neuron that's receiving the message, right? We're sending a message from the presynaptic neuron to the postsynaptic neuron, like, hey, somebody pinched my foot. Send that message along, right? The membranes face each other across a fluid-filled synapse. And as I said, the neurotransmitters will diffuse across the synapse. And the presynaptic terminal contains vesicles, and those are those containers, those little balls of neurotransmitter here. Okay. Here's some pictures. Right here you can see the vesicles in the neurotransmitter. And here are neurons that are all synapsing on another neuron. So although this is very simplistic, like it's one neuron that communicates with one neuron, in reality, there are many neurons all communicating with each other. And it's getting the neurons are getting many signals all at the same time. So what happens when an action potential is conducted down an axon? Several of those vesicles, synaptic vesicles, fuse with the membrane and spill the neurotransmitter into the synapse. We call this exocytosis. So here's another picture. Now we have neuron A here and neuron B here, and we're sending a message from neuron A to neuron B. So this is the presynaptic neuron, and we're sending an action potential, right? An action potential was sent down the axon saying, hey, I have a message. Here are these vesicles of neurotransmitter. And when the action potential gets to the end of the cell, or the presynaptic terminal, those vesicles start to move. They slowly move, and you can see, here's one, it fuses with the end of the cell, and the neurotransmitter spills out. Here's another vesicle that started to move through the presynaptic terminal. It fuses with the membrane, and the neurotransmitter, or the chemical, spills out into the synapse. And this process of releasing the neurotransmitter is called exocytosis. And these chemicals, these neurotransmitters, start to diffuse or to move across the fluid-filled synapse until they get to the postsynaptic cell. This is neuron B, the postsynaptic cell. And the postsynaptic cell has these receptors. That's what these blue markings are. They're receptors, and the neurotransmitters fit into these receptors, and basically that's the way a message is sent. This is, hey, I've got a message, and they send this neurotransmitter across. The neurotransmitter binds with the receptor, and now neuron B says, ooh, I got a message, and then we'll send an action potential down its axon. So the neurotransmitter, as I said, diffuses across the synaptic cleft and attaches to binding sites on the postsynaptic receptors. Those receptors are special protein molecules that are embedded in the postsynaptic membrane. And when those neurotransmitters, these neurotransmitters bind to the receptor, it basically activates the next neuron and the message is sent and it's passed to the next neuron. And I have some asterisks here because we're going to talk about it depends on the type of neurotransmitter. Some neurotransmitters are excitatory, meaning they're going to continue to send the message, while other neurotransmitters can be inhibitory, meaning they stop a message from being sent. But either way, that's the way a message is sent. And this is a chemical message. So we say that transmission of messages in the nervous system is an electrochemical process, an electrical and chemical process. The electrical process, right, is the action potential, and it's the way a message is sent within a neuron. And then the chemical part of the process is the way that one neuron communicates with the next neuron by sending these little chemicals, these neurotransmitters, to bind with receptors on the following neuron. So neurotransmitters, as we said, are chemicals that affect the electrical signaling of the receiving neuron. So we have a neuron here. Here are its dendrites, right, receiving messages. Here's the soma. Here's the axon. And if there's an action potential being sent, an electrical impulse, an action potential being sent down the axon to the presynaptic terminal. And here's a bigger picture of the presynaptic terminal that has the vesicles of neurotransmitters. And then when the action potential gets to the presynaptic terminal, those vesicles start to move and fuse to the membrane, and the neurotransmitters spill out, binding to the receptors on the postsynaptic cell. Now, there are different types of neurotransmitters. You may have heard of some of these: dopamine, serotonin, glutamate. And some of these neurotransmitters are what we call excitatory neurotransmitters, and those increase the chance that the postsynaptic neuron will fire, will send an action potential, and it will depolarize the cell. Remember, that's how we say when an action potential happens, the cell is depolarized. It goes from that resting potential of -70 up to +30 and then back down. But there are also some neurotransmitters that are inhibitory. Sometimes we need to stop a message from being sent. We can't just keep sending a message. We need to stop a message, or we need to slow down our breathing or slow down our heart rate or whatever it is. And so there are inhibitory neurotransmitters, and these decrease the chance that the next neuron will fire or send an action potential. And so these neurons actually hyperpolarize the cell. So they change the cell from the resting potential of -70 to maybe -75 or -80, making it less likely that this next cell will fire an action potential. Let's see here. You can watch this short video.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission. Most communication between neurons occurs at a specialized structure called a synapse. A synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neurons are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is about 75,000 nanometers wide. The neuron where the signal is initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. Each vesicle can contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an electrical signal called an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their contents into the synaptic cleft. Once they are in the synaptic cleft, neurotransmitters interact with receptors on the postsynaptic membrane. They bind to these receptors and can cause an action to occur in the postsynaptic cell as a result. This action may involve increasing the likelihood that the postsynaptic cell will become activated and fire an action potential, or decreasing it. Eventually, the neurotransmitter molecules must be cleared from the synaptic cleft. Some of them will simply drift away in a process called diffusion. In some cases, the neurotransmitter is taken back up into the presynaptic neuron in a process called reuptake. Once back inside the presynaptic neuron, the neurotransmitter can be recycled and reused. In other cases, enzymes break down the neurotransmitter within the synaptic cleft. Then the component parts of the neurotransmitter can be sent back into the presynaptic neuron to make more neurotransmitter.
Okay. So, these are the steps of neurotransmitter release. We've talked about some of this. We'll get a little more detailed now. We have an action potential that's sent down the axon of the neuron here. Here's the myelination, and we've got an action potential here. Once the action potential reaches the presynaptic terminal, or the end of the cell, calcium channels open, and calcium comes into the cell. Calcium is actually what causes these synaptic vesicles that are filled with neurotransmitter to fuse with the membrane and release this neurotransmitter into the membrane. And the neurotransmitter binds on these receptors on the postsynaptic membrane. And when those neurotransmitters bind, these channels open up and emit some kind of ion, whether it be a positive ion, in which case you'd be more likely to send a message to have this neuron send an action potential, or they could be negative ions, in which case that cell becomes hyperpolarized and less likely to fire an action potential. And so what happens once those neurotransmitters bind on the postsynaptic cell and the neurotransmitters open, the channels open, we get what we call postsynaptic potentials. We get changes in the electrical difference in the cell on the postsynaptic cell. And so how does the postsynaptic cell react to the action potential? It depends what kinds of ion channels are opened on the postsynaptic cell. If positive ions, which are typically sodium ions, enter the postsynaptic cell, the cell becomes slightly depolarized, right? Goes, maybe you're at negative 60, negative 70 as the resting potential, and then those sodium channels open, sodium rushes in, and you get a more positive or depolarization of that cell. We call that an excitatory postsynaptic potential, or an ipsp, right? Because it's starting to excite the cell, it's starting to get it more positive, closer to the threshold of fire and action potential. An example of an excitatory neurotransmitter is glutamate. So we can go back here. If this neuron was releasing glutamate, glutamate would go into the synapse, bind to this receptor, and glutamate is an excitatory neurotransmitter. So this is actually an ion channel. This ion channel would open, sodium would come into the cell, and this cell would become slightly depolarized, slightly excited. We would have an ipsp, an excitatory postsynaptic potential. Now, if the neurotransmitter allows negative ions, typically chloride, into the cell becomes hyperpolarized, and that is what we call an inhibitory postsynaptic potential, or an ipsp. Okay, so something like a neurotransmitter called GABA. So if we're at a resting potential, the ipsp is inhibitory. The postsynaptic cell becomes more negative. And if the cell becomes more negative, it is less likely to fire an action potential, right? And so I wanted to get us back here, right? If this neuron was releasing GABA, which is an inhibitory neurotransmitter, GABA would diffuse across the synaptic cleft, bind to the receptor on the postsynaptic cell. This channel would open and chloride would come in. Chloride is a negative ion. And this cell would become more negative, more negatively charged. So instead of the resting potential of -70, maybe now it's at -73, -75. It's farther away from the threshold of excitation for fire and action potential. And so we call that an ipsp, or an inhibitory postsynaptic potential. So the EPSPs and IPSPs are constantly happening on the dendrites and the soma. An individual excitatory postsynaptic potential generally isn't enough to trigger an action potential. It doesn't allow in enough sodium to hit that threshold. But what happens, it's the sum of many excitatory postsynaptic potentials minus the inhibitory postsynaptic potentials that must be reached must reach the threshold to send an action potential. So you can see that dendrites receive thousands of synaptic inputs all the time. They're getting inhibitory messages, excitatory messages, and basically the cell adds it all up. And if that threshold is reached to fire an action potential, then the cell will. So how do these messages add together, or what we call summate, or sum together? We have EPSPs and IPSPs that travel passively over time. And we have either spatial or temporal summation. So the EPSPs or IPSPs that arrive near each other, that's spatial summation, will summate together. And EPSPs or IPSPs arriving close together in time, that's temporal summation, will also summate together. So let's take a look at this, because I know it sounds confusing. Now we're at the postsynaptic cell. Here's our postsynaptic cell, and it's getting all these messages from all the neurons around it through the synapse, right? And the neurotransmitters are diffusing across the synaptic cleft and binding to the receptors on the dendrites of this postsynaptic cell, which are then opening ion channels. And what we have here is a cell that's at resting potential of -70, and it's getting a message from an excitatory message from this first neuron here, E1. And this is your excitatory postsynaptic potential. It depolarizes cell a little bit. It probably allowed sodium into the cell. Remember, sodium's a positive ion, but just a little bit, not enough to reach the threshold for an action potential. And then here's E2. This is another excitatory synapse, where this neuron is releasing another excitatory neurotransmitter, and you're getting a little bit of sodium that's entered into the cell. And here's another excitatory postsynaptic potential, but not enough to reach threshold. Now, let's talk about summation here. So here is temporal summation, which occurs when two EPSPs are produced in rapid succession, so shortly in time, close together in time. You get your first excitatory postsynaptic potential. One of these, here's a message coming in through E1, and then right after it, another action potential. So you get one excitatory postsynaptic potential here, and even before the cell can come back to resting potential, you get another quick postsynaptic potential, and they basically add together to reach this threshold of excitation, and then the cell will fire an action potential. Now, you can also have what we call spatial summation. This occurs when different synapses produce ipsps on the same postsynaptic neuron at the same time. So if you're getting an excitatory message from E1 and an excitatory message from E2 at the same time, so both of these synapses are letting in some probably sodium, then those two add together to increase or to depolarize the cell and reach the threshold of excitation, and then an action potential is fired. Now, you can also, this same cell can be getting inhibitory postsynaptic potentials. So you can also have some synapses that are letting in negative ions like chloride. And so all of this is summating together. We have an excitatory synapse that's letting in a little bit of sodium. Then we have an inhibitory, right, synapse that's letting in some chloride. This is hyperpolarizing the cell. And in this case, we're not getting to that threshold of excitation. So we have temporal summation. This is when you get two messages sent quickly over time that add up together to trigger the action potential. Or spatial summation, to get two messages at the same time from different neurons, right? But because they're both affecting the same postsynaptic cell, they can add up together to reach that threshold of excitation and trigger an action potential. All right, we've talked about that. And we have talked about this, the steps of the synapse, right? So the action potential reaches the presynaptic terminal, calcium channels open, the synaptic vesicles start to move and bind to the, fuse to the membrane, neurotransmitter is released and binds to the receptors on the postsynaptic cell. Now, we said those receptors then open channels that admit ions, maybe sodium for an excitatory postsynaptic potential, or chloride for an inhibitory postsynaptic potential. But now we have to get rid of the excess neurotransmitter that's in the synapse. So there's a lot of neurotransmitter that's released. Some of it will bind to these receptors, but some of it is not used. It's just kind of hanging around the synapse. And what we need to do is clean that synapse up to get it ready for a new message to be sent. So how do we clear the neurotransmitter from the synapse? There are two different ways. We can use degradation, sorry about that, degradation or reuptake. And so reuptake is right here. It's the idea, here is our presynaptic neuron. So our message is being sent like from the top of their computer screen down, and here's our postsynaptic neuron. Reuptake is a process by which if there's extra neurotransmitter, there are actually transporter cells in the presynaptic cell that bring back reuptake or kind of recycle the neurotransmitter in the synapse. It takes it right back up into the presynaptic cell. It won't get used again, but it cleans up the synapse in this way by taking up any unused neurotransmitter and bringing it back to the presynaptic neuron. And then another way to kind of clean up the synapse is enzymatic degradation, where there are enzymes in the synapse that take any extra neurotransmitter and break it apart. So you can see it, like cuts it in half. And once it's broken apart, it can't affect the receptors anymore. It's kind of useless, and it'll sort of become a waste product. And then, of course, some of it will just diffuse away from the synapse. But those are the 3 ways to clear up any excess neurotransmitter in the synapse. All right, thank you very much.